It is widely accepted in the gastroenterology world that microbiome diversity, the sheer variety of bacterial species living in your gut, is the single most important predictor of microbiome health. And probiotics are all the rage. Every day, I get patients in clinic asking me if I would recommend probiotics for them. So I spent the last 6 weeks actually reading the probiotic research, not the label claims, not the influencer posts, not the wellness blogs, the peer-reviewed clinical trials, the endoscopy studies, the meta-analyses, the actual research. And I keep coming back to the same uncomfortable conclusion. The supplement industry is selling most of you something that doesn't work. I'm Dr. Matthew Glover, and I'm a clinical gastroenterologist here in Las Vegas, Nevada. And I made this video because I am tired of watching my patients spend $60, $80, $100 a month on probiotic supplements while the actual science tells a very different story. So let's go through it together, all of it. The studies they're not quoting on the label, the findings that should have changed this conversation years ago, and what the evidence actually says you should be doing instead. Before we talk about what doesn't work, I need to spend a few minutes on why any of this matters. Because if you understand what a healthy microbiome actually looks like, everything else in this video will make a lot more sense. Your gut is home to somewhere between 38 to 100 trillion different organisms. We're talking bacteria, fungi, viruses, archaea, over 500 to 1,000 different species living in an ecosystem that co-evolved with humans over millions of years. And what makes a microbiome healthy isn't necessarily the quantity of bacteria, it's the variety, the diversity. Here's what diversity actually does for you and why it matters so much. A diverse microbiome produces a much wider range of metabolites, chemical compounds that influence almost every major system in your body. Short-chain fatty acids like butyrate, which literally feed your colon cells and maintains the integrity of your gut lining. Neurotransmitter precursors like serotonin. Over 90% of your body's serotonin is produced in the gut. Compounds that regulate your immune system, your inflammatory responses, and even your insulin sensitivity. When researchers look at people with inflammatory bowel disease, conditions I treat every day, one of the most consistent findings is a dramatic loss of microbiome diversity. Fewer species, less richness, less resilience. The same pattern also shows up in obesity, type 2 diabetes, autoimmune conditions, depression, colorectal cancer, cardiovascular disease, and in Parkinson's disease. Study after study in all of these conditions finds the same signal. The sicker the patient, the less diverse the microbiome. And it goes the other way as well. People who age the healthiest, the centenarians, the people in their 80s and 90s who are still sharp and physically functional, tend to have remarkably diverse microbiomes. Microbiomes that look more like a young, healthy person's than a sick person's. So when we say diversity is the single most important predictor of microbiome health, this is not a minor academic point. This is the central organizing principle of microbiome and overall gastrointestinal health. A diverse microbiome is resilient. It can recover from disruption, resist pathogens, maintain the gut lining, communicate properly with the immune system, and produce the metabolites your body depends on. And a low diversity microbiome, it's a microbiome that's struggling, vulnerable, producing fewer of the good things and potentially more of the inflammatory things. This is the standard that every gut health intervention needs to be judged against. Does it meaningfully increase microbial diversity? Does it make this ecosystem richer and more resilient? Keep all this in mind as we go through what the probiotic data actually shows. Let's start with the biggest picture finding because I think it's the most important and it's the least talked about outside of academic circles. In early 2026, a systematic review and meta-analysis was published in BMC Medicine, a peer-reviewed medical journal published by Biomed Central. The researchers did a comprehensive search across Medline, Embase, and Cochrane databases and identified over 9,000 articles on probiotics and microbiome diversity. They whittled that down subsequently to 47 studies that met rigorous inclusion criteria. Of those, 22 were randomized controlled trials with over 1,000 subjects that had enough standardized data to include in a formal meta-analysis. Their research question was direct. Does probiotic supplementation produce meaningful changes in gut microbiome diversity in healthy individuals? The answer, across every standard diversity metric they measured, the Shannon Diversity Index, KO1, Simpson's Index, was no. No statistically significant difference. Not across different durations of supplementation, not across different probiotic taxonomic families, not when they broke it down by risk of bias. No significant change in healthy adults. Think about what the industry actually claims their products are doing. Support a healthy microbiome, restore balance, more thriving bacteria. The marketing always implies more diversity, more flourishing bacterial communities, but the data says that's not what's happening for most people. And look, diversity isn't the only thing that matters, but it is the primary thing, the benchmark, the marker we have the most confidence in. And across 47 trials and more than 1,000 subjects, the signal is this. If you're a healthy adult, the probiotic capsule is not moving the needle. Now, let's talk about why probiotics may not be working, because the mechanism is fascinating, and as a gastroenterologist myself, this is the study that hit me the hardest. The foundational assumption behind taking a probiotic is intuitive. You swallow some bacteria, they travel through the GI tract, they set up residence in your gut, they do good things there. But almost all probiotic research tests this assumption by looking at stool samples. You give someone a probiotic, you test their stool, you find the probiotic bacteria in the stool, and you declare it a success. The bacteria made it through, they're there, study complete. Here's the problem with that, and it's something I care about deeply as a GI doctor. Stool bacteria are merely transit bacteria. They're bacteria passing through on their way out. What matters for your health, what interacts with your immune system, what produces the metabolites your body actually depends on, is what actually lives in you, what's colonizing the mucosal lining of the gut, the wall, the tissue, the actual resident community, not what bacteria is simply moving through you. A landmark study published in Cell, one of the most prestigious scientific journals in the world, did something almost no probiotic study had done before. They actually looked inside the gut. They performed endoscopies and colonoscopies to take direct biopsies of the gut mucosal lining, not stool, actual tissue. They took healthy volunteers, gave half of them an 11-strain probiotic for 4 weeks, and gave the other half a placebo, then scoped them and biopsied their gut wall directly. What they found is something the supplement industry doesn't want you to know. The researchers divided the probiotic group into 2 categories. They called them resistors and persisters. The resistors, and this was the majority of the subjects, showed essentially no mucosal colonization. The probiotic bacteria showed up in their stool, so if you had just run a standard stool test, it would look like it was working. But at the gut wall, at the mucosal lining where bacteria actually need to be, absolutely nothing. The native microbial community actively blocked the incoming strains. And here's what makes this even more striking. The researchers could predict which group someone would fall into based on their baseline microbiome composition. Your existing gut community determines whether a probiotic has any chance of working in the first place. If you have a well-established, robust, healthy microbiome, it is going to resist colonization from the incoming strains. Your gut knows who belongs there, and strangers don't get past the door. This is called colonization resistance. It's your microbiome acting like an immune system for itself. So here's the situation most healthy people who buy probiotics are in. You're paying $60 to $100 a month for a supplement where the majority of users in a direct biopsy study showed no mucosal colonization, where you have no way to know whether you're a resistor or a persister without a microbiome composition test, and where the large meta-analyses found no measurable change in diversity regardless. The industry is selling this to you as a done deal, but the science says it's a coin flip at best. Now, let's talk about the most common reason people reach for probiotics, which is after a course of antibiotics. Antibiotics kill bacteria indiscriminately, the bad bacteria causing your infection, and a lot of your good bacteria along for the ride. Gut microbiome is disrupted. Common wisdom says, take a probiotic to replenish the good bacteria. Your pharmacist has probably said this, and someone in your family definitely has said this. However, the research suggests something far more complicated. A second study published in that same issue of Cell, also from the Weizmann Institute, looks specifically at what happens to the microbiome during the recovery phase after antibiotics. They compared 3 groups, one that spontaneously recovered with no intervention, one that took a standard probiotic, and one that received an autologous fecal microbiota transplant, meaning they were recolonized with their own pre-antibiotic microbiome sample. The spontaneous recovery group, which was no probiotics, just wait and let the gut heal on its own, had their microbiome return to baseline in roughly 3 weeks. The probiotic group significantly delayed recovery. In some participants, the microbiome hadn't fully returned to pre-antibiotic baseline even after 5 months. 5 months compared to 3 weeks. The hypothesis the researchers proposed is that probiotic strains, by taking advantage of the cleared field that the antibiotics had created, were actually occupying the ecological niches that your native bacteria needed to recolonize. You've wiped the slate, dropped in some temporary tenants, and now your original community can't get back to the spaces that they need. This was a rigorous study using direct mucosal sampling, and it fundamentally challenges something that has been treated as conventional wisdom for years, which is take your probiotic after antibiotics. And this may be one of the most widespread pieces of gut advice that simply isn't true. The evidence does not support this. Okay, so if probiotics do not meaningfully increase the microbiome diversity or help after antibiotic recovery, then what does? Because I want to give you a real answer grounded in evidence that you can actually act on. And the most important study I want to walk you through is one that a lot of people in academia consider one of the most significant gut microbiome papers published in years, but almost no one outside of research circles is talking about it. It came out of Justin Sonnenberg's lab at Stanford, published in Cell, Host, and Microbe in 2021. They ran a 10-week randomized controlled trial comparing 2 interventions in healthy adults, a high-fiber diet versus a high-fermented food diet. The high-fiber arm got their fiber intake up to around 45 grams per day, roughly triple the amount of most Americans eat. We're talking vegetables, legumes, whole grains, the conventional eat more fiber for your gut advice taken seriously. The fermented food arm ate yogurt, kefir, kimchi, sauerkraut, kombucha, fermented vegetables, up to 3 to 6 servings per day. And the results were striking, and not what you'd predict based on most mainstream gut health advice. The fermented food group showed a measurable, sustained increase in their microbiome diversity, and their inflammatory markers improved significantly. There was a reduction in 19 different inflammatory proteins, including interleukin-6, which is a major driver of chronic inflammation and is elevated in everything from heart disease to depression to cancer. The high fiber group, despite tripling their fiber intake, they showed no significant change in microbiome diversity. Not a marginal difference, no change. That's a complete reversal of what the conventional eat more fiber for your gut advice predicts. Now, fiber is still important. It feeds your existing microbial communities, supports short-chain fatty acid production, and has clear benefits for gut transit and colon health. I'm not telling you to stop eating fiber, but this study does suggest that if your goal is to specifically increase microbiome diversity, fermented foods appear to be a far more powerful lever than fiber alone. What makes this even more interesting is something Justin Sonnenberg said in a New York Times interview after the study was published. Some of the new microbial species that appeared in the fermented food group could not be fully traced back to the foods themselves. He said they don't know entirely where the new species came from, which suggests that fermented foods may be creating gut conditions that allow other microbes to colonize or bloom. That's not just introducing bacteria. That's changing the entire ecological landscape of the gut so that beneficial species can take hold. This is a fundamentally different biological mechanism than swallowing a simple probiotic capsule. And the key detail on fermented foods, not all fermented foods count. Shelf-stable pasteurized sauerkraut, the kind you can find in a can at room temperature, the bacteria are all dead. It doesn't count. You need and want refrigerated sauerkraut with live cultures. Real kimchi, full-fat yogurt with live and active cultures confirmed on the label. Actual kefir. The Stanford study was running 3 to 6 servings per day to get the maximal effect, more than most people eat in an entire week. This is a serious dietary change, not a sip of kombucha once in a while. So beyond fermented foods, let's talk about what else the evidence supports to keep your microbiome healthy. Number 1, dietary plant variety. The American Gut Project, one of the largest citizen science microbiome studies ever conducted, over 10,000 participants, found that people who ate 30 or more different plant types per week had significantly more diverse microbiomes than people who ate fewer than 10. Not 30 servings of the same vegetables, 30 different types of plants. Herbs count, spices count, walnuts and almonds are different types, black beans and chickpeas are different types. This is achievable without completely overhauling how you eat. It's about variety, not volume. The mechanism is straightforward. Different plant fibers, different structures of polysaccharides, different ratios of soluble to insoluble fiber, different polyphenols feed different bacterial species. More variety in equals more variety out. You are literally cultivating a more diverse ecosystem every time you introduce a new plant type. And what else is beneficial for the microbiome? Avoiding what actually damages your microbes. This is unglamorous advice, but the literature here is real. The research on antibiotic overuse and microbiome disruption is well established, but there are 2 additional things that get less attention than they should, and these are emulsifiers and sleep. Emulsifiers are additives found in an enormous range of processed foods. We're talking carrageenan, polysorbate 80, carboxymethylcellulose. These are in salad dressings, ice cream, non-dairy milks, processed breads, and the research, including animal studies and some human data, shows that they can disrupt the mucus layer of the gut and alter the microbiome composition in ways that increase permeability and inflammation. The data in humans is still developing, but it's concerning enough to be taken seriously. In chronic sleep disruption, there are now multiple studies showing that poor sleep quality measurably alters the gut microbiome composition, including reducing microbial diversity. The gut-brain axis is bidirectional and real. You can eat perfectly and still have a struggling microbiome if you're chronically sleep deprived. For anyone who wants to go deeper on why probiotic responses are so individualized, the Institute for Systems Biology's Gibbons Lab published research in early 2026 showing that computational models can actually predict whether a specific probiotic strain will colonize a specific person's gut based on their baseline microbiome composition. This is promising technology, but it also definitively confirms what the evidence has been suggesting all along. One-size-fits-all probiotic supplementation is a fundamentally flawed approach. Your gut is not the same as your partner's gut or your parent's gut. A probiotic that might actually persist for one person will pass straight through for another. I want to be completely clear here about something because I'm not making a blanket statement that probiotics are worthless. That would be the same intellectual failure in the opposite direction. Taking a complicated body of evidence and flattening it into a simple narrative. There are specific, well-studied clinical situations where specific probiotic strains have demonstrated real, meaningful benefit. To be clear, certain lactobacillus strains for antibiotic-associated diarrhea, bifidobacterium for some irritable bowel subtypes, Saccharomyces boulardii, which is actually a yeast, not a bacterium, for C. diff recurrence. These are strain-specific, condition-specific, and mostly studied in people with actual gut dysfunction, not baseline healthy people who are trying to boost their microbiome. And in my own clinical world, for patients who have had a proctocolectomy, meaning surgery to remove the colon and rectum for severe ulcerative colitis, and developing pouchitis in the surgically created pouch. In this instance, the probiotic formulation VSL number 3 has a genuine, replicated clinical evidence for prevention and maintenance. That is a very specific product for a very specific patient population for a very specific indication. That is completely different from a healthy person buying a generic 50 billion CFU capsule at Whole Foods. The critical distinction, and this is what I want you to carry out of this video, is that the evidence for probiotics is condition-specific, strain-specific, and population-specific, not generalized, not universal. The supplement industry takes the evidence from these specific clinical scenarios and uses it to imply benefit for everybody. That's where the science and marketing part ways. So if the evidence is this weak for general probiotic use, why does this industry generate $77 billion a year and keep growing? A few reasons, and they're worth naming. Number one, the placebo effect in GI is enormous. This is not dismissive. The gut-brain axis is real, and expectations influence GI symptoms. An IBS placebo response rate in trials runs 30 to 40%. If you believe something is improving your digestion, you will often feel like your digestion has improved. The feeling is real, but it doesn't mean the probiotic did it. Number two, the regulatory gap is the industry's best friend. In the United States, probiotic supplements are regulated as food, not pharmaceuticals. They do not need to prove efficacy before going to market. They need to demonstrate safety, which most probiotics are, but they are not required to prove that they do what the label implies. If a pharmaceutical company wanted to market a drug that supports digestive health and restores microbiome balance, they would need years of rigorous clinical trials. A supplement company simply needs to not poison anybody. That gap is exploited brilliantly and systematically. Number three, most studies are measuring the wrong thing. If you fund a probiotic trial and measure whether your bacteria show up in the stool, they probably will. Now you've got a study that says clinically tested increases beneficial bacteria. That statement can be technically accurate and practically meaningless at the same time. A capsule that passes through without colonizing the gut wall will still show up in a stool test. The industry knows this. Number four, the people telling the truth speak carefully. Physicians will say, well, the evidence is mixed, and results vary by strain, and more research is needed. That's honest, and that's what the data supports, but it does not go viral. This probiotic transformed my gut, and here's my before and after. That always goes viral. And that asymmetric exchange is exactly why this channel exists. I want you and my patients to know the truth. So let me close with 3 things that you can do starting this week. Not supplements, not products, actual interventions with actual evidence behind them. Action number 1, add fermented foods to your daily diet. real ones, refrigerated sauerkraut, and check that it has live cultures. Full fat yogurt with live and active cultures on the label. Kefir, kimchi, miso. The Stanford trial was running 3 to 6 servings daily to get a measurable effect. Start with 1 or 2 servings and build from there. This is the single most evidence-backed dietary intervention we have for increasing microbiome diversity. Not a capsule, fermented food. Action number 2, start counting plant types for 1 week. Write down every different plant you eat. Vegetables, fruits, legumes, whole grains, herbs, spices, nuts, seeds. Count the types, not the servings. If you're under 20 by the end of the week, you found your biggest opportunity and leverage point. The target is 30 different types per week. And the American Gut Project data across 10,000 people suggests it makes a real and measurable difference. Action number 3, the next time you're standing in front of the probiotic wall at a pharmacy or scrolling probiotic reviews online, ask yourself 3 questions. What specific condition am I treating? What specific strain has clinical evidence for this condition? And what was the dose used in the research? If you can't answer those questions, and most labels won't help you, that tells you something important about what you're actually buying. If you have ongoing gut symptoms, we're talking about chronic bloating, changes in your bowel habits, blood in your stool, unexplained weight loss, iron deficiency, stop self-treating with supplements and see a gastroenterologist. These symptoms can be completely benign, but they can also be early signs of conditions such as inflammatory bowel disease or cancer that are very treatable when caught early and very difficult when they're not. All the research I cited is linked in the description below. Read it for yourself and be your own judge. If this video was useful, consider subscribing. I make evidence-based gut health and GI content every week. And leave me a comment. Are you currently taking a probiotic? Has anyone actually tested their microbiome diversity before and after? I want to know. I read every single comment, and your questions help determine what videos I'll make next. I appreciate you taking the time to watch, and I will see you in the next one.